Next, we'll move on to bacterial transduction. What is bacterial transduction and how it is going to have an advantage in the field of science. Now, transduction is a process from which the bacteriophage or the virus is going to infect the bacteria and kill the host cell and is necessary for horizontal gene transfers in bacterial cell. So we'll learn how about the discovery of transduction, how to define the term, the process of mechanism of the horizontal gene transfer. Now there are two types of transfer, that is generalized and specialized. So we'll learn about them as well. So this is bacterial transduction, wherein I have my bacteria and my bacteriophage is going to infect. So now I have my horizontal gene transfer. Now we already know what is horizontal gene transfer. We know what is vertical transfer, wherein there is transfer or inheritance of genetic material from the parent um, to the offspring set. Now this process is going to effectively generate clones of the parent. But in horizontal gene transfer, this is going to have this in this the bacteria is going to have the ability to transfer genetic material from one cell to another cell. Now, this process will allow genetic recombination or mixing of the genes of two different DNA pieces to form a DNA sequence which is unique. So, in bacteria, this process is very crucial for genetic diversity in the population. Now, in the environmental conditions are constantly changing, the organisms also need to adapt to these changes to survive. So, using this genetic recombination, this will generate the genetic diversity wherein the organisms is now adapted to the environmental changes. So then I have the mechanisms wherein this horizontal gene transfer will take place. Again, we have the three, that is transformation, conjugation, and transduction. So here we'll start with the transduction. Now, we have heard that the video or image is going to go viral, right? And nowadays, if anything that is having a unique thing shown in the video, then it goes viral. So why does this come as viral, right? Where does this metaphor come from? It comes from nature, wherein the viruses are going to be the uh, organisms that have the ability to invade it, invade the host, hijack the cellular machinery, and force it to build a million copies of virus. So these copies are then released so that they can attack the new host through the population. So these viruses that infect the bacteria are called bacteriophages, which literally transfers to bacteria eater. Now, sometimes instead of just infecting the and hijacking the host, the virus picks up and transfers some of the host DNA, and this is called transduction. Now we have the two types of transduction, right? The first one, we have generalized transduction. So in this case, bacteriophage interaction with the bacteria is very simple. The bacteria is going to invade, hijack the host, copy the DNA or copy its DNA, and then kills the host by popping like a balloon or lysing the cell. Now the new baby bacteriophages are going to infect the host cell. So what exactly is happening? I have my bacterial cell with its nucleus. Then I have my bacteriophage that is invading it. and this is going to sell its DNA to it, and there will be incorporation of this DNA. And next I have many copies of this bacteriophages in it that are building, and these will pop out and kill the bacterial cell, right? So this, in this process, the bacteriophage chops up the host chromosome and makes smaller fragments of them. Then when these baby bacteriophages are going to get assembled, some of this host DNA chunks that was there will accidentally get packed in the new viral particles. So they, there you can see there is mixing of two DNAs, and then the new host cell where they are going to inject, there will be recombination of DNA. So that was our generalized transduction. Then we have specialized transduction. In this, instead of just hijacking and killing the cell, the viral DNA goes dormant. Okay, uh, this dormancy is going to come after incorporating itself into the bacterial DNA chromosome. So the host will survive and continue to grow and divide. By this, there is going to be passing of the incorporated viral DNA, and whatever offsprings are created of the bacteria will have this viral DNA in it as well. So the bacteriophage reactivates itself. If, because it was in dormant phase, it can reactivate itself and cut open the host cell and move out. Now then it can hijack the cell to produce newer bacteriophage, lyse the host cell and infect other cells. So this is how the specialized transduction takes place. Now the trick here is the virus cuts itself out of the host genome. It takes that chunk of DNA with the, it takes little bit of the host DNA along with it. Now once the bacteriophage infects the new cell, the old host DNA that was tagged along will genetically get recombinated in the new host DNA. So each type of virus inserts into the host genome at the same place at the same time. So when these viruses are going to cut themselves free from the genome, they will typically take the genes that are found right beside their insertion point. Now, this is unlike general transduction where random genes are picked up, right? Here it is a specialized spot or specialized sequences. Now, how did this transduction even get discovered? How did such tiny organisms were studied? So, in 1952, there were scientists who were working on the mutant cells of Salmonella species. Then they noticed that the genetic recombination was taking place. They had they had to rule out the various mechanisms of horizontal gene transfer. First, they ruled out transformation by verifying that the culture was free from contaminated DNA, right? Since there was no contamination, there was no way the bacteria is going to take up free floating DNA. Then they ruled out conjugation by keeping the cells isolated from each other in a thin membrane. When the cells couldn't touch, then they couldn't conjugate. So when they still couldn't find anything and still detected genetic recombination, this was the time they found out there is bacteriophage particles in the culture which was conducting the transduction. So we have aerobic bacterial metabolism. When we begin with bacterial metabolism, we'll study the definition and process. Now, it is very important to understand the metabolic process of individual blocks and how they are going to generate energy. So here we'll study the metabolic pathways of aerobic bacterial metabolism. So we'll start with metabolic building blocks. Now humans also consume food for energy, use oxygen for respiration, and this is called metabolism. But bacterial cells also carry out exactly the same processes. So understanding the bacterial metabolism can be difficult. There are many pathways and enzymes, and it's not always clear how to link them together. So in this lesson, we will use a simple way to visualize metabolic pathways. So here we are going to see some basic pathways, how they are going to fit in a bacterial cell. So one of the easiest way to visualize the connection between metabolic pathway is to imagine that there are various pathways that are going to be the individual blocks. Let's use this build to build a metabolic model of bacterial cells that are going to grow in aerobic condition. So what is this aerobic condition? Here the compound that is going to be used in the process is oxidized. So using oxygen as the terminal electronic acceptor. Now that doesn't make sense, but in the end of the lesson you will get to know what is happening. So we'll start building. So now our cell is going to use the glucose as our energy source under oxygen condition. So this is a simplified way of all the building blocks in the cell of aerobic respiration. And we are going to now start with sucrose. And we need to go up till here. So when we start with glycolysis,